Yeah, thanks for inviting me. Let me first check the people who are online. Can they hear me? Can somebody unmute or give us a thumbs up or something like that?
Yes, we hear you.
Excellent. Okay, thanks. So please give me questions as I go along. You don't have to wait for the end. So I'm Kasper Hansen. I'm an associate professor at Biostat at Hopkins, and I really like work on applied statistics and particularly statistical genomics, which I've been doing for quite a long time. So I know you're all experts in that here at Michigan, or at least some of you. So I'm going to talk about something that's like really single cell. But let's first talk about what are other stuff that I'm doing right now. Let's see. There was something about hiding the controls, right? Hide floating. Okay. So in the next couple of weeks, my lab is going to post 2 preprints that could be of interest to people here. I've already talked about both of them with the highly enjoyable conversations I've had so far. One about RNA velocity. If you know what it is, you know what it is. It's been sort of a hot topic in single cell biology. We've been spending a lot of time wondering about whether it makes sense. And we are adding sort of our thoughts on this, which complements a couple of other papers of similar flavor that has been published here in the last, or been put on bioRxiv here in the last couple of months. And we have yet another perspective on it. And then we are finishing up a paper on a framework for detecting selection acting on epi-alleles such as DNA methylation or histone modification in human promoters. We have an earlier version of the work that we posted on bioRxiv a long time ago, but now we have a new and improved version. The findings are sort of more or less the same, but we have some more findings. But we have a much more, I would say, perhaps rigorous sort of population genetics approach to like why we're doing what we're doing. That was definitely missing in the first version. I almost chose to talk about this stuff here instead of the cell cycle, but anyway, I picked the cell cycle. Okay, so this is really a work that was published in Genome Biology not that long ago. It is led by C.G. Singh, who's a postdoc of mine, who is phenomenal. And we've done it in collaboration with Laurel Goff and Genevieve Sten O'Brien. She's on the job market this year. And it's been super enjoyable. It's sort of a little light here in the darkness of the pandemic. But we have had it's really like the highlight of the week is sort of our weekly meeting here. One of the highlights of the week, but it's definitely a highlight. So you can find all the stuff in this paper here, but I'm going to give you like a description of what it is we're doing. So first, not everybody on this call may be, living and breathing single cell data. So I would say if we take a step back and we think about beyond genomics, what am I really doing in this work here? So this here is sort of a practical example where we develop a method for sort of fishing out a very specific signal in a complex data set that possibly have multiple like very different signals going on. Like, so we're trying to extract a very specific component, and we want to go in and extract this component. We don't want to kind of find it. We just want to say, let's grab the component and fish it out. And then this is also an example of a method that really works, what the machine learning people call auto distribution. And that really just means it works in very different data sets. We have we have some internal controls in this work. We've discovered that you can basically evaluate whether or not it works in any data set you apply to. And we have applied it to, I guess, by now, easily 100 different data sets. It's always worked. And that's pretty unusual because let's be frank here, right? I mean, at least in my little part of the world, I develop methods for genomics. And, I like to say that they're good methods, but, when we apply them on a new data set, we cross our fingers and we hope that it works. Like, it definitely doesn't always work. It's pretty unusual you have something that just always works. An example of one of the few methods I would say in genomics we have that, quote, always works is Steve Horvath's work on predicting calendar age from DNA methylation data. I don't know if people here have tried to run it themselves, but it's totally amazing. You take his lockdown predictor, there's no refitting, there's nothing, you just pump in your own data into it, and it just like completely works. I mean, it is amazing. I really feel that leaving aside sort of the biological implications, which are quite big for this piece of work, this really deserves to be studied for like, how did he come up with this predictor? Because it is extremely unusual to have something that always works. So out of distribution, right, is sort of this idea that the machine learning people, or this word the machine learning people have labeled on it, where you write a paper, you take some data, you develop some deep learning model or some neural network or whatever, you evaluate, you write your paper. So the step here in the beginning, when I say data model fitting and evaluation, I mean the whole paper, right? The research is sort of done. You publish it, you declare massive success. You know, you have found a new machine learning method that works, and you have now solved the problem. What we often see is that we then move to a different data source. It's perhaps the same type of data, but you get data from in a different context, and we run the machine learning thing, and it just doesn't work. And you know, that's a big problem for the practical applications of prediction data. And in machine learning, they're sort of internalized a little bit. They've kind of said, well, it's because the new data we have a different distribution than the old data. So why would we ever think that the model that we have developed and the procedure we developed to maximize some prediction, minimize some prediction error, should work on a new data? Okay, but I mean, I'm going to say I think this framing is a little bit dangerous, because this is really what we want to do, right? I mean, the example we really want to do is come up with some kind of prediction method that actually works on other type of data. If it just works on the data you have on your laptop, most of the time we don't really care about it. Okay, so here's an example from machine learning. Oh, I can minimize it. So back in 2017, Andrew Ney, who is a big machine learning guy at Stanford, published a bioarchive paper, an archive paper, where they tried to predict pneumonia from chest X-rays. So they have X-ray images, you have a ton of them, and they want to diagnose pneumonia. This is not a problem of high practical importance. When you diagnose pneumonia, you typically use more information than just to check the chest X-rays. But they were like into deep learning. They developed a deep learning model, and they declared, you know, massive success. And in fact, this is him on Twitter some years ago, basically saying that, you know, in 5 years from now, you probably heard this, radiologists are like out of the job. Their computers are better than radiologists. And you know, they claimed, or the doctor who was involved, one of the doctors who were involved in this basically claimed that this was super interesting, because this was the first time that they actually barely outcompleted humans. Now, when you read the paper carefully, you can say they just had 4 people that they benchmarked against. But you know, it was pretty interesting. It was widely reported, massive headlines. Then the paper was actually never published. Instead, they published a revision of their method that is slightly different in PLOS Medicine. But anyway, that was great. I can also say that a quote that I didn't come in here was that they did all of this work in 1 month, which, you know, the doctor here was pretty impressed by. And I'm like, that's probably not a good sign. So in 2018, a group, I forgot where they come from, led by Sekia, at that time, he was a resident in radiology. So he was like a doctor doing this stuff. And he was really interested in deep learning. So he was like, this is amazing. Now we have the computer that can do this. So they developed, they took a team and they started to apply this model and test it. And what was different from their thing is that they had access to 2 other large-scale data sets with chest X-rays and pneumonia. So you could take the model and you could fit it, or you could both fit it or run it with the fitted model on the new data. And the new data does not work. The model does not work as well on the new data. It worked really well on the Stanford data. It did not work on new data. And it was actually kind of complicated to understand why, because they did all of these kind of experiments. It's pretty interesting to read it, where they tried to, what happens if we fix the model, but we retrain it on the new data, or we retrain it on a combination of the new data and the old data. And by and large, this shows that the model didn't actually work. It is not portable outside of Stanford. So In 2021, Andrew Ney was interviewed for an IEEE publication and was substantially more humble. That he says here that, you know, we can actually do this really well when we get data from Stanford, but it doesn't work when we work down the road and we get data from a different hospital system. Which is, you know, that was definitely not what they said back in 2017. And this is sort of what we mean by auto-distribution thing, right? They have data from a different source, they have different scanners. Perhaps the scanners are calibrated a little bit differently. The study population is different, et cetera, et cetera. And this thing here that humans can just do, we can't actually do with this model. I mean, perhaps there's other contexts where we can do it. Okay, so this is a bit of a cautionary tale. So I think this is a huge problem in prediction. I think it's also a problem in genomics. In genomics, we're a little bit more, you know, relaxed about it. The way I think of it is like you take a model, you take an approach, you get a new data set, and you kind of hope that it works, right? And you know that it won't always work. Okay, so in this work here, we're interested in the cell cycle. I'm going to talk a little bit about the cell cycle. But the first thing I'm going to say is that when we started working on this, it was clearly a well-developed area. There was like 7 different methods out there, at least, probably more, that all declared, you know, massive, overwhelming success in doing the cell cycle. It's sort of a well-studied area. So let's talk a little bit about how hard the problem is. So for those of you who are not familiar with single cell and all the buzzwords, I'm going to say that I think of like 2 main systems we could be interested in. And the first thing is you have a bunch of cells in a petri dish in the lab. They're just dividing happily. They're sort of in a steady state, and they're just proliferating. And in such a system, the only thing that really goes on, there's no environmental signals, nothing is happening, the cells are just dividing. Almost all the variation in that data set is explained by cell cycle. That's, of course, not what we're really interested in when we study cell cycle. In reality, we typically want to take a tissue sample or a more complex system where other things are happening, differentiation, environmental exposures, et cetera, et cetera. There are many different cell types, and that's where we really want to get the cell cycle. So the sort of the cells in a dish, nothing is happening. It's like really easy, and the many ongoing processes is really hard. That's sort of intuitively clear. We also have different technologies. So it turns out that the method we have developed here works actually across all technologies we've tested it on. And let's just say that the single cell data differs a lot in sort of the amount of information content per cell. C1, which is sort of an older technology, has very high information content per cell. And then as you go down to the droplet-based methods, you lose information content per cell. And then when you go down to combinatorial indexing, you know, the stuff developed by Shendu and Trapnell up in Seattle, the information content per cell is like really bad. All of these methods tend to trade off number of cells with information content. So you're not going to get hundreds of thousands of cells on a C1 machine, but so you have sort of this trade-off. So in general, when you're estimating something at the single cell level, I mean, of course, it's easier when you have more information, right? So it's going to be, generally speaking, easier to do this on C1 data than on combinatorial indexing data. The problem we have in this field here is that all the gold standard data is the easiest possible data. It's data that's coming from a C1 machine, and it's data where we are profiling cells where cell cycle is the only driver of expression. So all of these methods here actually do pretty well on the gold standard data set. It's not so clear to say whether or not one method does better or worse on the gold standard data set because it's pretty easy. Most of them fail miserably when you get into more complicated things. Either it's a little bit method dependent, but you know, a combination of less information content and more complicated signal make these methods fail. Okay, so what's the basic idea we're doing here? Okay, so we're going to start off by, now I'm going to use a couple of fancy words, it's pretty simple. We're going to construct an embedding that represents cell cycle using a reference data set. We do this one time, and then we fix this reference data set. Okay, that's a key aspect here. We're just picking a data set we like. We've generated ourselves. We have 2 samples in the data set, so it's not exactly like a massive amount of variation. And And there will be a lot of questions, or we had a lot of questions when we started doing this. Is this like the only data set where we can do it in? And why shouldn't there be some reference data sets that works better for some, data sets or not? But in practice, this here just works well for all 100 data sets we've ever used. What we're really doing is we do PCA in this reference data set. And this reference data set, the only thing that goes on is cell cycle. And I'm going to show that when the only thing that goes on is the cell cycle, you can just use PCA to extract the cell cycle thing. Now, when you do a PCA and you take the first two principal components, you're estimating, a two-dimensional linear subspace. And we have, from basic linear algebra, we have a projection operator or orthogonal projection operator that can project new data into this two-dimensional space. So what we're going to do when we get a new data set is that we're going to take the new data, we're going to project it into this fixed linear embedding, and then we get the cell cycle, right? Really super simple. We're just fixing the weights in the PCA, so to say. What we're doing is we do a little bit more. We do a PCA, and the PCA is going to show up as a circle. I'm going to show some data on that. And then we're going to use the angle of the data point of the cell around the origin to estimate the cell cycle time. So our cell cycle time is going to be a number from 0 to 2 pi, represents sort of the degrees around it. And we are going to show in practice that when we project new data in, we also get a circle, and we take the angle. At a very high level, this approach was suggested in a publication from my co-authors in Cell Systems. They suggest there that you should estimate reference embedding and project new data into reference embeddings. I gotta say, when I read this paper, I was not very convinced by it. It was sort of more, I was like, who says this is gonna work? But now I've used it for this particular problem with the cell cycle, and I'm like convinced this is the way to go here. So I'm also gonna say, you always, when you develop new methods, you're sort of a little bit skeptical that it works. And I remember when we started working on this, we played a little bit around with it. It was not kind of at all clear that this here would be great. And then, over time, we applied it to more and more data sets. We applied it to like 5 or 10 data sets as part of early work on the paper. You just, in the beginning, you sit down, you look at the plots, and you're like, well, does it really work this time? And then when you've answered affirmative to that in many, many data sets, your belief in this sort of just increases. It's really hard to impact. But we've used it in many, many data sets now. It has never failed. Okay, so let's talk about the cell cycle. Okay, so the cell cycle is a fundamental biological process. It's highly conserved. It's a continuous process. It's tightly regulated. We know that deregulation of the cell cycle is basically associated with many diseases, and it had a catastrophic impact on the cells. The time, the wall time, or clock time it takes to complete a full cell cycle varies greatly between cell cycles, between cell types, even within an organism. And if you go and you look for cell cycle genes, you're going to find a lot of them. If you take the cell cycle term in the gene ontology, we get around 1,700 genes. Now, there's other ways of getting a list of cell cycle genes, and there's a huge variety in like what is called the cell cycle gene. Okay, so the cell cycle is usually represented as this circle here, and I've drawn it in the opposite direction of how it's normally being depicted, because by mistake, that's how we oriented our circles when we did our methods development. That's a big mistake. We definitely got criticized by it later on, but then it was too late to change it. Okay, so a normal cell hangs out in what is known as G1. G here stands for gap. And that it's called gap because it's not part of the cell cycle. Okay, so this figure here is really focused on the cell cycle and not on like the reality of the cell in the context. Because if you take cells, they spend by far most of their time in G1. And they hang around and they do whatever the cells are doing. And then at a certain point in time, they decide, okay, now we replicate. And this is a pretty well understood process. You go through a replication phase where the DNA inside the cell is doubled. So this is something you can see under the microscope if you have a big enough microscope. But you double the DNA. That's the S phase. Then you go through a growth phase called G2, that's short. So the cell gets bigger and the amount of RNA in the cell goes up. Okay. And then it's become big, it has a lot of RNA, and the cell goes through cell division. So the two copies of the chromosomes move to like separate ends of the cell, and the cell gets split down the middle. And the RNA and the protein in the cell gets distributed between the two daughter cells. So if you take it from a sort of a linear point of view, you first have, most of the time being spent in this G1 phase. Then you go through the S phase where you double the amount of DNA. Then you go through the G2 phase where you kind of increase RNA up to some steady state. I think it's about, you basically double the amount of RNA. And then you go through the M phase where the RNA gets cut in half because it gets divided into the two daughter cells. And then, very important in practice, we then have two cells, right? So most of the picture you have up here is all about, in textbooks, it's all about the cell cycle, and they tend to kind of, don't focus so much on the fact that after M phase here, we have two cells. Now, a little bit of lingo. So G1 is, as I said, the time where the cell is doing what it's doing, but there's also a phase, or there's a state called G0, which happens sometimes. Some cells are terminally differentiated, and they decide we are not going to divide anymore. And then they go into a phase called G0. We don't really have in biology any good markers for the difference between G0 and G1 that is independent of cell type. There may be markers for it in specific cell types. I don't know, but there's it's a deep biological problem to differentiate between cells that are going to cycle again sometime in the future versus cells that have stopped cycling. It's also a little bit unclear whether or not cells in G0 are ever going to cycle again. Perhaps they just decide, I'm not going to cycle for the next 10 years, and then I'm going to cycle. So in some way, philosophically, you can think of G0 as just a really long G1 phase. There are probably some people who are studying senescence and so on who are going to come after me when I say that, but sort of, we don't really have, and this is going to be important for this talk, we have no way of differentiating G0 and G1. So I'm going to say cells that are actually cycling are stuff that are in the process of the cell cycle. That means cells that are either in M, G2, or S phase. And then cells that are not cycling are out in the G1 phase. People sometimes include cells in G1 as actually cycling cells because they're going to cycle sometime in the future. Okay. So in general, why are we interested in this problem? So in general, how a cell goes through cell division and cell cycle is pretty well understood. It's super fundamental. We learn about it in high school. We learn about it again in university. And it's super important, right? Of course, biologists have spent tons of resources understanding this. Now, there's also this link between cell cycle and differentiation, right? So I draw this as sort of a spiral thing. So here we have one cell. We go through the cell cycle, and then after the cell cycle, we have two cells. And then you have these sort of lineages. You go through the cell cycle again. You split out into two cells, right? So in many cases, we have some belief that there's a link between differentiation and cell type specificity and the cell cycle. There's this concept in biology called precocious differentiation, which means it has sort of problems going through the differentiation hierarchy. And often that's linked to some measurable defects in cell cycle. So we think that there's something about cell cycle and differentiation, and we don't understand this at all in biology. So we are really interested in understanding this link here between cell cycle and differentiation. I'm not saying we understood that at all. We're taking the first step towards exploring that. In many single cell analysis, you are really interested in understanding the cell cycle as a confounder. And that's because now we're measuring individual cells. Some of the cells are going to be in the cell cycle, some of them are not. And there's a lot of gene expression that goes over the cell cycle. Okay. So as I said, there's a lot of existing work. A lot of the existing work doesn't really predict a continuous cell cycle. They predict a certain number of stages, which for some, if you, for example, want to fish out cells that are actually cycling, you don't need anything more than like a sort of a rough idea, like is it cycling or not. These two methods, we really like conserved. It's like one of the best methods for sure. These two methods just label each cell with 3 different labels. Then we have swapper here that is a new method. It doesn't work that well, but we're going to use it in some of our downstream plots, some of the plots on the later slides, because it labels things with higher resolutions. You have 5 different states instead of 3 different states. That's the only reason why we're using it. The plots we're going to show are going to be a little bit noisy because it kind of sucks, but it's going to have higher resolution. Then there's a number of continuous time predictors, OSCOPE, PICO. PICO is from Matthew Stevens' lab, ReCAD, Revilio, who all tries to do some kind of continuous time prediction. But for example, ReCAD here predicts sort of an ordering of the cells. It doesn't link the cells directly to time. Anyway, so and all of these methods work pretty well on well-behaved data sets. It's not hard to take a cell cycle or to take a single cell data set of these sun signals and find cell cycle inside. What's hard is to do it automatically. Okay, so let's start with some exploratory data analysis. Okay, so this is the data set that we're actually going to use as a reference data set, but let's just say it's a data set here. We have 12,000 cells. We look amongst the 1,700 genes that are labeled as belonging to the cell cycle from the gene ontology. We take the 500 most variable genes, we mean center them, we don't standardize variance, and then we make a PCA plot. And we get something like this that I'm going to describe as an ellipsoid. We clearly have a donut with a hole in the middle. And then I'm going to foreshadow a little bit. We're going to basically assign cell cycle time by putting a center here at 0,0. If you sit down here, you're going to wonder why 0,0, why not minus 5,0? We're going to get back to that a little bit later. We're going to pick 0,0, and we're just going to measure the angle. So the color of these points here is this swapper stage estimation that tells us that if you look at the colors and you think a little bit about it, there's sort of a lot of mislabeling going on down here. So ignore that for a moment. We basically have this progression here with S phase, G2, G2M, and then all the cells that are here are sort of in G1 and G0. So the fact that you get a circle when you do a PCA plot in some data sets has been observed many times, including in this very famous paper by Alter in 2000 in PNAS. So people who are old, or even older than me, will remember that one of the first big microarray papers that ever came out, they did the cell cycle. So it's hard to do cell cycle with bulk measurements, but there's a way to chemically synchronize cells so all the cells in the disk are at the same stage of the cell cycle. Once you have that, you can just do bulk measurements. So there's a big Nature paper, I think it's a Nature or Science or something like that, who cares, where they basically started the cell cycle using microarrays. And then in this paper here by Alter, and I think it's Botstein, who's the last author, they took the same data and they did a PCA of it. This paper is widely cited as the paper that introduced the singular value decomposition in genomics or in microarray data. It's cited like 10,000 times. They introduced this concept of like the eigengene, etc. And they basically show that they get a circle. Now, a circle seems totally sensible, right? We are studying this phenomenon that kind of goes in circles. When you look at any textbooks, you see a circle because you have this closed loop. But why do you actually get a circle on this PCA plot? That was not completely obvious to us. So we did some math, which is a little bit unusual for me. I'm typically more practically applied, but we did some math. Okay, so we did some simulations where we do something super simple. We have some Gaussian noise, but we basically say we have a bunch of cosine functions, and we're gonna have the functions are gonna have different amplitude, and they're gonna have a single peak because we don't have any like scaling factor in front of the time. They're going to have a single peak. They're going to have a different location and different amplitude. So here's 2 genes. Okay. And then we do a PCA of it, and we get something that's a circle here, and we can explain that really well. So if you look at sort of methods from functional data analysis, you can basically show that the mathematics of this circle here is entirely determined by the eigendecomposition of a 2x2 matrix that is determined by sort of the set of amplitudes and locations. And then, and you can show basically that because of that, there's a strong relationship, and there's a one-to-one relationship between where the peaks are and this sort of cell cycle time. Now, the killer thing in practice in single-cell genomics, right, we don't have that. We don't have, we are trying to order the time points so they look like this. So we have data that looks like this. We don't have a time order. Now, because we're doing discrete observations of a continuous process, and every single gene in a given cell is observed at the same time point. That's sort of that's a given when you do single cell measurement. The PCA of this data here and the data down here is exactly the same. Okay, that's what makes this work. So what we are doing over here is we are taking this data here, putting it into this thing here, and estimating the angle and reordering. And it works perfectly. This is probably a phenomenon that works under many more complicated types of periodicity. But when we are looking at cases that just have a single peak, it's particularly simple. We get a circle in the first 2 dimensions. And that's all we have to think about for the cell cycle. I think that there's a little interesting paper about what do you do if the functions you have multiple peaks on them, and how do you sort of align them and so on. And when you do simulations, you can see you don't necessarily get a circle in the first 2 dimensions, but you probably get some kind of regular structure in low dimensions. You have to identify that and so on. But we haven't worked that out. But we worked it out for a single peak perfectly. Okay, so we will say this here is a decent model of the cell cycle, because the cell cycle genes that we are interested in have a single peak. Now, that's what we see here, although there's a certain element of circular reasoning here, because on the x-axis on this plot here, on the y-axis, we have the 500 genes. On the x-axis, we have our inferred time order. So there's a certain amount of circularity here. But you can basically see that every single gene has a single peak, or it's not even all the genes that have a clear peak, but the genes here with the red stuff here, those are kind of the 2 clusters here, cluster 2 and 3. And then we have sort of these things here that are sort of more wild. Okay, now in this plot here, we're doing exactly the same thing, but for a different data set we had lying around. Both data sets have more than one sample, and that's pretty important. We do this data integration thing where we extract shared sources of variation within the data set. And that really helps us hone in on the cell cycle. It really gets rid of some sort of sample-specific variation. Both of these data sets are sort of brain-related, but otherwise, these are completely separate analyses. They don't see each other. There's no sharing. So we get basically the same circle, although the shape of the circle is a little bit different. And over here, we're just looking at the weights of the PCA, the 2 different PCA in the 2 different data sets. And this is the first PC. There's a massive Pearson correlation coefficient. And all the red dots are genes that we know are super important for the cell cycle. Top 2A, anyway, they're super important for the cell cycle. So we really kind of get the almost same coefficients in the 2 different data sets. No sharing of information or anything, right? That really tells us to some extent that we're seeing the same thing. Now in this data set here, we always have it oriented so the G0 and G1 phase are out to the out here to the right. And that is something we don't get from the math. The math just tells us here is the origin in this simulation thing. It just tells us the origin is somewhere on the circle. This here comes because of a PCA and properties of expression along the cell cycle, because the first dimension is always going to be the difference between G0 and sort of this G2M that you have down here, which is basically the biggest expression difference in that. That's really nice because it makes it really easy to transfer cell cycle time between data sets. We don't have to rotate anything. It just comes out. The only thing we have to think about is flipping the sign of the eigenvalue. Okay, now this here is a pretty important thing. So whenever you have a cell cycle time in continuous time, you can take your favorite gene, here's 3 of my favorite genes, and plot the expression dynamics as a function of inferred time. Okay? And what we're seeing here is 2 data sets overplotted on each other. It's very discrete data because it's single cell data. We have done a lowest smoothing. It is definitely not the best smoother of this data, but we didn't really have energy to come up with something better. This is sort of good enough. And we can basically see that the peak locations are the same between data sets. We've done nothing to harmonize them. It's completely separate analysis. And they peak kind of where we would expect. So we have these genes that we know quite a lot about from biology. And we know that they're going to peak. They peak at sort of the right places of the circle, given what we would expect based on this Schwabe estimator here, where out here we have sort of G2 starting on G2M. Over here, we have total UMIs. So total UMI is just the total number of reads after we filtered out UMIs, duplicate UMIs in each cell. That has been suggested to be a proxy for total RNA content. Okay, so remember I told you, after M phase, you should see a twofold drop in RNA content because the cell divided into two. So you can look at these two circles here. This here is sort of M phase. And you can look at the difference between the top, the lowest and the highest part of the curve, which is, you can sit down and do it a little bit more precisely. It's exactly 1 on the log2 scale, which is exactly twofold, right? So that fits perfect with our expectations, with sort of our understanding of the physicalities of this process. We're not the first people who have seen this, but it really tells us we are really getting something that tells us that this is definitely related to the cell cycle. Now, are there other time orderings that's going to give us the same kind of rough dynamics? For sure. Right. Okay, now we take a data set that fails, right? So these first two data sets were data sets where we have some cells in a disk, nothing is going on. The only thing that goes on is a cell cycle. This is a more realistic data set. It's a differentiation system in the mouse pancreas. And if you do sort of, if you do sort of the standard UMAP, you get a plot that basically reflects the expected biology. So in this system, you have ductal cells that differentiate into endocrine cells and then becomes sort of terminally differentiated cells. And this is exactly kind of the trajectory you would expect based on biology. What's interesting here is that it's really these two cell types over here that are actively cycling. The other cell types are terminally differentiated. So there is some relationship between this UMAP and cell cycle, but it's not really cell cycle as we had in the other. Now we do the same thing over here. We take a PCA and we take the top 500 most variable genes that are labeled as being cell cycle genes. And we get a PCA plot that really looks highly similar to the UMAP. So first of all, it is clearly not a closed topology. It's not a donut. Secondly, it really tells us that even though we had restricted our data analysis to the cell cycle genes, we see basically the same structure, right? That tells us something about that these cell cycle genes are not just doing the cell cycle. They are also doing other stuff. Like if it was the case that we had like 100 genes, the only thing they did was the cell cycle, they are orthogonal to anything else going on in the cell, this problem would be super easy. But that's not the case, and this is why it fails. Right? Whenever you have data sets that are complicated, you are going to have a PCA plot that doesn't show up as circle, and you're not going to get the cell cycle out. And a lot of these competing methods I talk about, they essentially start off with saying the PCA plot has to be a circle. Okay, now over here, I do kind of the same thing. So there's circles hidden in here. So I take all of the green cells, which I know are cycling. I only take those cells. I do a PCA plot of those, and I get something that looks pretty similar to the other plot, except there are many fewer cells, right? So, yeah, I have the conclusions here, right? We can recover on the left side by restricting to a single cell type, and we see that, basically in some cases, cell cycle genes participate in other biological processes, and we don't get a circle in the PCA plot. Okay, so what do we do here? Okay, so we learn a reference embedding, which really just means we do PCA. And we fix the projection matrix for the PCA. We have two samples. We have integrated them using, the integration module in SIRAC3. And this really helps remove noise. We are focusing on the shared variation between these two replicates, and that really filters out a lot of sort of sample-specific noise. We could probably do better if we had 10 samples, for sure, but there's a huge difference between one sample and two samples. Then we select our 500 genes, and we do a PCA. And then we take new data, we just project it into the fixed reference embedding. We take the new data, we mean center it, we log transform it, we correct for library size. Otherwise, we don't do anything. We put it through the linear transformation, and we compute the angle. Okay, one thing that I forgot to mention over here. So that I think is actually quite important. So we have these two data sets here, and we showed that the PCA is pretty similar. But even though in both data sets we picked the top 500 genes, there's only 300 genes that are shared between the two data sets. Okay? And this here is something that also makes a problem hard. In any given data set, it's data set dependent what genes are sort of above the Tricycle level. Okay? So anything where you take a reference data set and say, these 5 genes are super good for estimating cell cycle, you run into the risk that in your new data set, those 5 genes are not present. So you really have to deal with this thing here that is highly data set dependent, how many genes, which genes are present, even in 2 very well-behaved data sets. Okay, so here in the middle, we have the pancreas data set we looked at before. That was the one with the sort of the U-formed trajectory. And when we do this thing here, we fix the reference embedding, we get this beautiful loop-like structure. And when we look at the dynamics of top 2A, which is my favorite cell cycle gene, it basically peaks exactly where the other things peak. They peaked at around pi. Now you can see this data here, right? It's kind of crappy data. You have tons and tons and tons of zeros. It's highly discrete. I mean, it's because we correct for library size, so that's a little bit self-dependent. But I mean, it's not like high quality data, but it works. And when we look at it on the UMAP, this was a UMAP we had before. We can see this thing that I said that there's cycling cells up here and a little bit here, but over here there's nothing. Right? We can also see that in some cases, here's a HeLa data set, where in this case here, the UMAP is not a closed loop, right? It's sort of the cell cycle kind of goes from here to here, but it doesn't loop around. So this is sort of the problems that in many data sets, you don't necessarily get these sort of circles, right? This here is the other data set. The data set here in the middle, but projected into the other data set. You can see that it changes the shape, right? Here, you can see that it changes the shape. It looks more like the other data set. And we've done a fair amount of experimentation that basically tells us that the shape of the PCA is really determined by which genes are included in the PCA. So if we take the 2 data sets and we do a PCA of them where it's only the genes in common that we keep around, they look much more similar. Okay, if you really like RNA velocity, which I don't, but if you really like it, this here just kind of confirms what we see. RNA velocity analysis confirms what we see with what we kind of inferred other ways, right? We see this sort of circular pattern around in the PCA plot. Now let's look at some gold standard data sets. So here is a gold standard data set where they use the Fuji system that's considered the best system for measuring cell cycle. Now, it is the best system for measuring cell cycle, but it has some problems when you use it with single cell expression data. So what happens in the Fuji system is you engineer the cells so you can track the protein levels of 2 key cell cycle genes through imaging. So you can take a microscope and you can track the cell and the protein level of those 2 genes, of those 2 proteins continuously through time. And for sure, that's the best way of measuring cell cycle time. But it requires that you take a cell and you put it under the microscope for like 24 hours. What happens in single cell data is that you take your cells, you take a 1 snapshot of them, and then you send them off for sequencing. That 1 snapshot is just way more noisy than having sort of the continuous curve of the individual cell over time. So down here you have the 2 cells. You have the 2 proteins, and you define something. These papers, this paper here defines something called Fuji time. You have this sort of not closed loop. And this loop here is there's really a loop that goes between the bottoms down here. And the reason why it's separated out in this plot here is because this here is a key, this is a protein that's involved in a key checkpoint. So during a cell division, this protein has to be erased from the cell for to continue. So what I'm trying to say is that from here to over here is a couple of minutes in real time, but it just has to be degraded really fast. Over here it's pretty slow. We're going to see, we're basically going to see that this future system has really high resolution here, and it's not as good in the rest of the system. So over here, we take our tricycle position, and then we cheat. Let me emphasize we cheat. We take all of these cells here that are inside this box here are here, and we move them over here. So what we've done is we have rolled out the cell cycle time. You can see here is the period just after 0.9 pi, and this is also the period right after 0.9 pi. And that's exactly this checkpoint where Fuji can really tell you very precisely, are you on one side or the other side of this checkpoint? And with the expression, we just lie. They're sort of roughly around the point. We don't know if they're left or right. Now, so how well do we do? Well, we could then take, so now we have two different measurements of cell cycle time. So we can take our favorite genes and we can fit a lowest line and we can say, what's the R square? How much expression variation do these two time concepts explain? That's what we do over here. We have the R square for Fuji time, we have the R square for tricycle, and basically for every single gene, we have a higher R square for cell cycle, for Fuji time. And that's because our method, we think, works way better in the rest of the cell cycle. We kind of suck around this little point inside the transition I talked about, but the rest of the time, we're actually better than Fuji. Because our method is based essentially on the expression of 500 genes, and they have a noisy image measurement for 2 genes. But down here, we are doing the same thing, but with a different experiment did by Matthew Stevens and Jörg Gillard, where they basically the same experiment, except they analyzed a little bit differently. So we would actually argue that based on this, that we believe, may not agree with us, but we actually believe that the tricycle time is at least as good as Fuji time when considered over the entire cell cycle. You can also do fact sorting, where you kind of isolate cells at different parts of the cell cycle. We also have great results on that. We have a lot of, I think we have 36 supplemental figures in this paper, so there's going to be a lot of stuff where I'm just going to be a little hand wavy, and then you can look in the paper for like the 50 different pictures we have. Okay, so existing methods do well on simple data sets. Basically, they all do well on simple data sets, but sometimes they fail catastrophically. In the case where they strongly disagree with tricycle, we have some evidence presented in the paper that tricycle performs better. We've never seen tricycle fails, and we have sort of this caveat, right, that comparing methods based on the gold standard is a little bit biased because it's really easy. Now, we also know why a lot of these methods fail. Like, I mean, sometimes it's simply based on like what they do right. I mean, this, as I said, this recat method, okay, I'm not going to go back to it, but this recat method starts off by essentially making a PCA plot and then using the placement on the PCA plot as sort of cell cycle time. That is only going to work if the PCA reflects cell cycle. If it doesn't reflect cell cycle, it fails catastrophically. So RAT that predicts sort of 3 different stages actually works surprisingly well in our hands. It's pretty robust. Okay, so here I've been pretty interested in the shape of the PCA plot. So here is to the left is sort of the plot you've seen, and we are basically doing a PCA with this plot here, where we change which genes goes into the PCA plot. Right? So on the left we're using 400 genes. On the right, we're using 50 genes, so you can see how it changes shape, right? You can also do experiments where you thin the data and so on, and so part of how much noise you have in this donut really depends on the sequencing depth as well. So if you have data that is really poorly, that is sequenced very lightly, and you make this PCA plot, and you make this sort. This is not really a PCA plot, right? Because it's a projection into this reference embedding. You're gonna see like a pancake and not a donut, because you can't see the whole because of the increased noise. But you can take, so we were very much in doubt whether or not that worked when we looked at that, but then again, you can look at the dynamics of individual genes, it works pretty well. Okay, so I would say that fixing the projection is really central for handling these multiple cell types and new data. We are not discovering the cell cycle inside new data, we are fixing it and just getting it from new data. I really think that one of our key aspects that makes this very adaptive to new distributions is that we are using this angle between the two principal components. So if you have two data sets that are sequenced at very different depths, for example, both principal components are going to go upright, but the angle is sort of going to stay the same thing. We are also, for some reason, really robust to drop out of these genes. So a lot of people in the cell cycle world have spent time saying, can I predict cell cycle using as few genes as possible? And in fact, if you look at this plot here, you might say, why is the idiot using 500 genes? Why is he not just using like these 20 genes down here? Right? They're the ones with high coefficients and so on. So we have played quite a bit with that. Don't have a good graph to show it. But basically, every time we kind of optimize it to a data set, we lose generalization ability. It's really, if we drop down, if we go from the top 500 to the top 100, we do great in some data sets, but in some data sets, we start to lose ability to do this. So it's really important that we have like all of these cells here, all of these genes here, and we are sort of a little flaky about what we're using, right? So if the gene is not expressed in the new data set, it's gonna not contribute to the PCA thing. And that apparently seems to be okay. It's also really important when we do all of this that we start off with a data set with at least 2 samples and we do some data integration to remove sort of sample-specific noise. Okay, so we have a couple of interesting questions. So we really want to take this into comparisons. And all I've done now is estimating cell cycle time. This is a data set from the developing mouse retina, and it's ordered in order of like basically days or time points during mouse development in the eye. And you can kind of see that the width of the cloud is really time dependent. And it sort of seems to move. First it's small, then it goes up, then it goes back again. Down here, we have fully differentiated cells, right? And you can see there's like not a full terminal differentiated cells. We have nothing out here, right? We really think that there's something about the shape and the size of this cloud here that tells us something about cell cycle length, because we know that cell cycle length changes through here. I match this up with the width of this thing here. We haven't really figured that out yet, but we're interested in it. We hope that there's something we can figure out. We have no hidden tuning parameters. There's nothing like, it's just a linear, it's just that we just do a linear, we just do a matrix multiplication, we do an angle, no tuning parameters, nothing, and it just works. There's a little bit of variation. If you really want to compare across data sets, you can see sometimes there's a little bit of variation in the peak location. And we think we could probably fix that with some form of normalization. So we're quite interested in moving this into a comparative analysis, and we don't, but we really want to caution people against doing that right now. When we look at replicates, especially, we don't feel that replicates are as close together as they should be. So they both clearly, you clearly get the cell cycle time, but it's not like down where we really want to do. So one solution to that is just like come with 100 replicates. And then I'm sure we could do this. But we have like, in most applications, we have 3 replicates. So we're not at 100 yet. We've also seen that there's some data sets out there where the replicates, for some reason we don't fully understand, are completely crazy reproducible. Like the noise between replicates is so little. And then there's other data sets where the noise is high and we have no idea why. So we're trying to figure this out. We haven't done it yet. Okay, here's an example where Tricycle kind of fails. We got I'm almost done here. We got a GitHub issue from Luke Sapir, he's a guy in Europe, and he was like, hey guys, what the hell is wrong? You can see here in the color thing, right, that the plot here is centered at 0,0. And that's it. He was like, what is going on here? These cells are not supposed to differentiate. So basically what happens is that he is in a system that is not really, there are very few different, this here is a circle of cell cycle. What happens is because we mean center the data, we are subtracting the mean, and basically because all the cells are in G0 and G1, they basically become mean 0 and the origin of the thing is here instead of say around here. Now, this seems really simple to fix. We spent quite a lot of time fixing this. It takes 2 minutes to diagnose it. You look at this plot and we immediately see what goes on. It's very easy to convince yourself that what I just said is actually true. It is crazy hard. So I was like, this is going to be easy. Just got to figure out is all the cells in G0 and G1. So, I mean, that seems like a really simple problem. And we have not been able to solve that problem because we always end up with having to make kind of technology-specific cutoffs and dataset-specific cutoffs. Because, again, it's very different from dataset to dataset. When is a gene expressed? When is it not expressed? Et cetera, et cetera. So it's easy to diagnose. It's easy to fix. Seems to be, we haven't been able to automate it. We also didn't spend months on it. But so anyway, it's super scalable because we're just doing a multiplication. You could run this on millions of cells in minutes. It always fails in our thing. We have this internal control, which is really incredibly nice. Now, the internal controls doesn't tell us that it works perfectly, but it does give us some kind of confidence that we are doing some right thing. It's generalized across data sets. I didn't really talk too much about it, but you've seen data now from multiple species, so we're just doing it across species. It doesn't just like, okay, I'm not done it across all species. I'm pretty sure that there's some species that's not going to work. I think so. But human and mouse, sort of mammals, it just works. It works across platforms. I mean, I've very rarely seen something that just works across platforms with nothing. No rescaling, nothing. Needs a small tweak when we apply it to a data set with almost no cycling cells, but that's really easy for an expert. And then we have this ongoing work about like, how do we use this to make comparison? And then also the thing that started all off, like, how can we use this to sort of study the interplay between cell cycle and differentiation? Okay, so that was my talk. Thanks for listening. So I know I sound a little bit like somebody who has bought the Kool-Aid, but I'm a little bit like, I've worked on genomics in many years. It's really quite rare that I found something that I can just apply to the data set and works. And of course, perhaps there's somebody who's going to email me and say, it didn't work in my data set. But I mean, we haven't seen that. We have used it on a lot of data sets so far.
Because I'm not working on this area, I have a very naive question. So all the examples you have given are all on the healthy cells. So I was wondering, is it possible to use a method on the disease cells, for example, the tumor cells? So in the tumor cells, for example, will they also have the similar cell cycle pattern as the healthy cells?
I had a little bit of a problem hearing. So you're saying that all the cells I showed are what type of cells?
So I was just wondering, will the cell cycle be different between the healthy cells and the tumor cells?
Oh, healthy cells and tumor cells. Yeah, I mean, you can use this on I mean, in our experience, we used it on some cancer data sets. It works beautiful. Like, if you like cell cycle, we had a recent paper in Nature where they did some stuff with cell cycle and some drugs and so on, and they did like crazy amounts of experiments. We have run it through Tricycle and we get something that's like, first of all, we believe the data because we agree with them. We got to the point where we really believe what comes out of this. But yeah, we can just do it like computation in like 5 minutes instead of spending 6 months in the lab. Now, okay, I mean, I'm not tested all cancer contexts, right? And I know some cancers are truly amazingly messed up. So I mean, but we have used it in cancer data sets totally successfully without any problems. And again, I think it really comes down to this angle, right? I mean, so you have, but if you have like, specific chromosomes that are heavily magnified, now I don't know, but we have used it in cancer data sets and it seems to work well to us. But I would be interested if you see something to the something that doesn't chime with that.
Thank you.
So I had a couple questions. In your early plots where you showed the Schwabi labels,
yep.
And it seemed like there was a lot of misclassification between, I think it was G2 and G1. Right,
Yeah, so you can basically, this method does not work very well. It's a newer method. We use it because it has higher time resolution. I mean, it has higher. Basically, you can't see it because of overplotting, but if you do the density plot of every single of these labels here, it's bimodal. There's a bunch of cells it gets right, and the cells that it gets wrong gets placed down here at the G0, G1 phase.
I see.
And then you can ask, how do I know it's G0 and G1 and not just us that are full of it? But you can look at other things that really strongly suggest that they're misclassification. In general, they have lower expression. They do seem to have do seem to look good, but they just have lower expression, they have higher rates of missingness, all stuff that because of how the method works will make it likely to be put into that category. So you can see very clearly G2 here, right? But I mean, basically you have a more or less equal cloud size of all of them. We have a huge set of figures that explores this in great detail in 10 different data sets in the paper, but I'm quite confident after we've done all of that they are wrong in a very specific way. They're wrong in the sense that some of the things that they classify to G0 down in what we think of G0, G1, they classify as being at a specific place in the cell cycle.
I see. So my other question, you sort of alluded to this at the beginning of like, you put the origin at 0, 0. If you were handed this plot with no labels, you would probably put the origin right in
so I mean, the
middle.
first thing, and this is sort of hand-wavy thing, but the first thing is it doesn't matter that much. Okay. You can move the origin around, doesn't change the time that much. Of course, it shifts everything a little bit, but it doesn't have a big impact. The other thing is that I'm kind of nervous about anything where we have to like learn it from the data. Right? I think this is just so beautiful because everything is fixed. And you know, this data set is easy to place, right? You can place it visually, right? But I mean, some of the data sets are a little bit more complicated, like something like this data set, you don't see a middle because it has 100,000 cells. Other data sets, you have pretty poor sequencing depth. These are, I mean, we have done a lot of work on these combinatorial indexing cells, and they're just like, the information content is really poor. It's very hard to kind of see the middle. And I'm just like, you know, it works. And we have to trade off a possibility for overfitting versus doing something like statistically appealing. And I mean, I don't think it has a big impact. I'm really nervous about overfitting. Right? It's a little bit sort of the same flavor as the can't you just do this with 100 genes? Although we know with the 100 genes, you sort of do worse. Yeah, I mean, so that's sort of a little hand wavy. For sure, I mean, and what you're saying is kind of, you know, it's well taken in the sense that the place where it works, where it fails, right, or sort of semi-fails.
Your GitHub issue is.
Yeah, that is where I didn't put it. But in that GitHub issue, I mean, with this data, I'm just giving me a good, I mean, it's going to be really hard to say it goes down.
So that mRetina data set that you were just pointing at,
yep.
That's one where if you moved the origin, you would reclassify a lot of cells because you have like a dense.
Yeah, to some extent. I mean, it does look, it's this data set here, right? Here we have it faceted, right? Yeah. And you can see that there are a couple of time points where it's labeling, but there's still quite a lot where, you know, this here is always going to be G0, G1, this here is so on. So yeah, I mean, there's going to be a little bit of misclassification for sure.
Okay.
And I'm also like, I mean, you know, absolutely, the worse the quality of the sequencing is, the less I'm going to trust any individual cell. I mean, that's how it has to be, right?
I'll let Laura ask your question.
Hi. So when you talk about the number of UMIs, can you give a sense of how many UMIs would be good and bad? So we have, you know, maybe between 1,000 and 10,000 UMIs per cell. When do you start to get concerned? I'm
never actually really concerned. Like when we take the single cell combinatorial indexing, we typically have something like on the order of 300 or 400 UMIs per cell, which is pretty pathetic. And it works there. All that happens is you do not see a hole in these plots here. So when you do you just see a cloud. And you look at the cloud. When I first saw it, when we did it the first time, I was like, the cloud looks really bad. But then, we did the QC we have done, we looked at the dynamics of top 2A, we looked at the UMI level and so on, and it works pretty well. I mean, but of course, there's going to be a higher degree of misclassification when you have those UMI. Like basically, it's the same thing Gene says, right? I mean, if you have a really clear hole in the cloud, you are much more confident about every individual cell, right? If you don't have a hole in the cloud, there are going to be some cells where if it's like, is it in the top part of the circle or the bottom part of the circle? It's like, and basically that's unavoidable, right? So I'm not really concerned. I think it works well on the combinatorial indexing stuff, but strictly worse than on the C1 data we've seen, right? I mean, there's no like free lunch here, right? I mean, if you're worse data, you're gonna have more uncertainty in your assignments, right?
Thanks.
If you said a 5,000 to 10,000, that's plenty. I mean, that's a lot.
Yeah, we've thresholded somewhere. I don't know what we went to, either 500 or 1,000 at the bottom, but we have what I'm curious is it really depends actually on so this is single nucleus data, and it really depends on what cell type it is as to how much data we have. There are just really big differences between cell types.
Yeah, and that's probably, I mean, I'm not saying I totally understand. So I'm claiming here in this talk that total UMI is a proxy for total RNA content. Other people have suggested that. I think the fact that it goes down twofold on our dynamic plot is a pretty strong indication that's true. Does it make total sense? Not completely to me. I mean, I think it has something to do with the fact that if you're more RNA to begin with, you're just going to have more RNA that survives all of the application process and so on. But I mean, we know different cells have different highly, I mean, that could be highly variable in total RNA content, right? So I would expect that it's partly explained by that. I mean, basically cell size.
Thanks.
Yeah.
So how sensitive are these results with respect to choice of your reference data set?
Yeah, good question. I mean, I'm going to say that, we have not taken 10 different reference data sets and done this. I mean, it's a fair, you could do it. What we have played a little bit with is the two data sets we started off playing with. So these two data sets I showed in the beginning. Let me go back here. These two data sets here, these two mouse sort of brain-related data sets. And there's not that big of a difference between doing it with one or the other. Do I think it's going to be better? I think you would probably get better results if you had a bigger data set, higher quality, more replicates, and you did it all. I'm not sure that it's going to matter that much. I mean, there's just going to be some intrinsic mislabeling and error in this coming from the fact that you're poor information content per cell, right? And we haven't really explored it. What I can say is that when we started this, we were really like, we have to use, it has to be dataset dependent, what reference data set we're going to use. We're going to find that this is a brain data set, it's going to work in brain, but it's going to suck for immune cells, right? That was what we thought. And that doesn't seem at all to be the case. So we've applied it to, so one of these single cell combinatorial indexing things was like one of these atlas things where they basically have done tons of different organs in the developing human. It seems to work pretty well everywhere.
So what should be the most important criteria while selecting an alternative reference data set? What's your suggestion about that?
I would never pick an alternative reference data set if I was going to analyze data. I mean, if I was going to do that, it would be because I'm going to sit down and spend 3 months just picking a better data set, right? I don't think we're going to do this anytime soon. Perhaps in a couple of years where we have a really large data set. But we really want a data set where cell cycle is the main driver, whatever that means. And then we would want to go through all the QC stuff, right? But I'm not like the little experimentation we have done with it doesn't strike me that this is not just like a ton of work for relatively little payoff. But perhaps, you know, if we do it in 2 years, and I might sing a different tune, right? I mean, intuitively, you should be able to do better with more replicates, I would say, and cleaner data, right? I mean, that should intuitively be the case, but how much better? That's not clear to me, and I've labeled it under not worth my time right now.
Thank you.
Yeah.
Have you thought about how you would get out something like a confidence interval or a region
No.
or something, right?
No, I've not thought about it. I've not thought about that. I think that's a great question. Like, I mean,
Cool. Other questions?
I mean, one thing that we are missing here, that we got a little bit of pushback from the reviewers, including Matthew, is that we're just saying, hey, we look at our favorite genes and it looks good, right? Can't you do this sort of quantitatively? The problem again comes back into this, that it's all measures are highly data set dependent. So if you do, for example, an R-square for all the cell cycle genes, if you have a data set that's really lowly, have really low UMI, most of the cell cycle genes are not going to express. You're going to have 0 expression, and your R-square is just going to be like 1, right? So you make a box plot of the R-square, and it looks pathetic. But then, you know, there are some in there that looks really good, right? It sort of comes down to this thing that I feel is sort of unanticipated, a little bit unanticipated once it matters. But I mean, how hard it is to make anything having to do with expressing cutoffs and so on, and have that work in a portable manner, right? So we actually spent, because we got asked to it by the reviewers, some time trying to develop an automated measure. So we would have some QC measure and was higher than, say, I don't know, 0.5, it would be like declare victory, right? And we basically gave up because we didn't feel that we could make something that really captured it. Yeah.
So do you think that this ports well because cell cycle is so universal? Or do you think that, are we only going to get well porting machine learning classification methods for stuff like cell cycle, or I guess the methylation age example is sort of a.
I mean, I don't. Yeah, that's a great question, especially given my introduction. I don't really know. I can say that, you know, if you're looking for something, if you're looking for a signal, it better be there, right? And absolutely, it helps that it's sort of similar. I mean, it's the same system, right? Having said that, as I've also alluded to a little bit, the actual expression level of the genes are different. And many of the cell cycle genes are involved in other things, right? That comes back to this thing here with like the PCA plot looking like this, right? And I think part of it has to do with this list of 1,700 genes. I think that we have some genes we know are doing core cell cycle stuff. And then we have a lot of hangarounds, like genes where you did some cell cycle experiment, you saw that it changed expression during the cell cycle, and you declared to a cell cycle gene, but it's perhaps just a passenger gene that was sort of upregulated as part of cell growth. Now, the logical answer to this is, well, concentrate on the 5 genes that we know are like clearly cell cycle genes, right? And I'm just like, we can't get it to work that well when we do that. It's also, you look at this plot here, right? And you're like, what are we doing with all of these cells up here? It's just like noise. I mean, but if we move them, we don't do as well. They're sort of used for anchoring the G0, G1 fate, the G1, G0. That's how I'm thinking about it. So we've done it. So you can absolutely do this with very few cells in a specific data set, but we lose the ability to generalize. So, well, if you want to generalize something, you better have sort of the same type of signal, right? I mean, it's going to be really hard to do prediction or infer structure if it's not there, right? And for sure, it helps. Like, I mean, the thing we really want to move into is sort of differentiation now, and that's going to be a mess because differentiation is not going to look like this. I mean, I think the simplest thing is to say, let's take the data and somehow subtract the cell cycle and hope that what's left is sort of differentiation. But that's going to be, you're not going to be able to have a reference space for differentiation. We may have a reference space for differentiation in a specific context, right? like Loyal is really into neurobiology. So perhaps, we can do this for, maturing neurons or something like that. But it's not going to work in like a different organ, I think. So yeah, I don't know. Thank you.